So now let's look at riboflavin or vitamin B2. Vitamin B2 is also called as riboflavin. This is the basic structure of riboflavin or vitamin B2. This portion over here is a pyrimidine ring. The B portion is the azine ring, and the C portion over here is the benzene ring. So chemically it is 6,7 dimethyl 1,9-D ribityl isoalloxazine, which is our riboflavin or vitamin B2. This is the basic structure of vitamin B2, and it comprises of these three parts. That is the pyrimidine ring, the azine ring, and the benzene ring. Let's see what is the chemistry of this vitamin B2. It is an orange yellow compound. So if you have a compound of vitamin B2, then the appearance will be orange yellow of that compound. It has a ribose alcohol, that is D-ribitol, and a heterocyclic parent ring structure, isoalloxazine. So the heterocyclic parent ring structure is the isoalloxazine. The one carbon, that is the first carbon of ribityl group, is attached at the 9th position of the isoalloxazine nucleus. So the first carbon of the ribityl group is attached to the 9th position. This one, 9th position of the isoalloxazine nucleus. Now let's see or have a look at the stability. It is stable to heat in neutral acidic solution, but not in alkaline solution. It is not stable in alkaline solution. Aqueous solutions are unstable to visible and UV light. The reactions which take place are irreversible. Riboflavin undergoes reversible reduction readily in the presence of catalysts to a colorless substance called as leucoriboflavin. So this is the colorless substance called as leucoriboflavin. So the riboflavin, and in the presence of light, it forms lumichrome in acidic and neutral solution, and in alkaline solution it forms lumiflavin, which is a yellow pigment, and it is soluble in chloroform. So as we have already mentioned that aqueous solutions are unstable to visible in UV light, and the reactions are irreversible. All the higher plants can synthesize riboflavin. In nature, it occurs both as a free form and also as a nucleotide form or as flavoproteins. Human beings and animals cannot synthesize this vitamin B2, and hence solely depends on the dietary supply. So we human beings and animals are fully dependent on our diet for this vitamin. In man, considerable amounts can be synthesized by intestinal bacteria, but the quantity absorbed is not adequate to maintain the normal nutrition. Hence, there can be a deficiency if this vitamin, that is riboflavin, is not supplied to us through our normal diet. Flavin nucleotides are readily absorbed in the small intestine. So as we have seen, most of the vitamins, the absorption of most of the vitamins takes place, or by far all the absorptions take place mostly in the small intestine. The free riboflavin undergoes phosphorylation, and this process, that is of undergoing phosphorylation, is a prerequisite for the absorption of flavin in the nucleus, in the small intestine. So when there will be formation, or when this riboflavin will undergo phosphorylation, then only there will be absorption carried out in the small intestine. Excretion is mainly in free form, up to 50% as nucleotides in the urine. Daily urinary excretion is 0.1 to 0.4 mg, that is 10 to 20% of the intake. Riboflavin is secreted in milk, 40 to 80% in the free form. Free and nucleotides tend to remain quite constant, 500 to 750 microgram daily, largely from the unabsorbed bacterial synthesis. So this amount is excreted through the feces. Let's see the occurrences and the food sources of vitamin B2 or riboflavin. The plant sources. High concentration occurs in yeasts. Appreciable amount is present in whole grains, dry beans and peas, nuts, green vegetables. Germinating seeds, example, grams or dals, are also very good sources of riboflavin. Animal sources are liver, kidneys, milk, eggs, carb meat. It has high content of riboflavin. Milk also has riboflavin to some extent, which is an animal source. What would happen if there is a deficiency of vitamin B2? There is no definite disease entity in case of deficiency of vitamin B2. Deficiency is usually associated with deficiencies in other B vitamins from the B complex. In human beings, lesions of the mouth, tongue, nose, skin, and eyes with weakness and lassitude is reported. There can be redness or shiny appearance of the lips. Then the lesions at the mucocutaneous junction at the angles of the mouth leading to painful fissures are characteristic of deficiency of vitamin B2. There can be painful glossitis, the tongue assuming a red, purple, magenta color. So that is the end to vitamin B2. Next we shall have a look at vitamin B3. What is the basic structure of vitamin B3 and how the vitamin B3 is metabolized and what are the various sources of vitamin B3. That's it. Thank you very much.